This is Dr. Vaughn Mancha, and this is an episode of Epigenetics in less than a half an hour. As we start off with our first slide, you'll notice that there's a GIF on the left-hand side, and what it's showing is how our DNA is wrapped around what's called nucleosomes, and that there are little tags on the nucleosomes called histone tags, which are epigenetically or above the DNA modified. And it's these little epigenes that tell the DNA how to package up or not. So in moving along to this next slide, this is where I really got interested in epigenetics. It was an article by Nature Journal, which is considered the Bible of evolution. And several people were coming up and saying, does evolutionary theory need a rethink? Well, I thought this was astounding that they should even think this way, because I thought it was supposedly in stone for the last 100 years. In fact, I went through a degree in biology in the late 70s, and that was the consensus then that evolution and Darwin were unapproachable. If you notice, they show the chillid fish, which are up in the Alaskan area, and that they have a different morphology, even though their DNA is completely the same. So how could these different forms be showing up with the similar DNA? And they've discovered that this is epigenetics. It's not mutations and random selection, as Darwin would say. This is basically modern evolutionary theory on how things occur. It's all enwrapped in what's called the central dogma. The central dogma is accurate on one level, but it's also been found to be inaccurate. The central dogma was basically made up by Francis Crick, and it says that the DNA transcribes to the RNA, which then transcribes to the protein. Now, this is true on a certain level, but there's much more complexity going on here that we've discovered in recent years. However, it it combines with the mutation theory of neo-Darwinism. So the mutations occur on the DNA, and then the RNA is made, and then a different protein is made. And subsequently, this new protein makes a bigger muscle or stronger bones or makes you better looking. And therefore, natural selection acts out on this. So this is the model that has been proposed for the last 100 years. And it's very simple and very appealing in a lot of ways to a lot of people, but unfortunately, this is very wrong. So in the example here with this GIF, we show a DNA double strand being mutated, and a new nucleotide is put into place. Then going back to the central dogma, the DNA is transcribed to the RNA, which is translated into a protein, which makes you a better person. And this is an example. We have a stronger penguin as a result of this mutation, and it's going to have more babies and populate the area. This is a very lulling, simplistic model, but this is basically what was proposed for the last 100 years. And it wound up lending to a lot of what we call just so stories. That just so was a term that was used by Roger Kipling in the 1800s in Victorian England. And, the evolutionists could make up stories that said, well, this mutation did this, and therefore they were going to, in time, they would wind up being this. Well, they never really proved that. They did prove that mutations can occur and then it can change the proteins to some extent, but never to the level to make whole new beings. And on the right here is a kind of an argument from the absurdum to that theory, when they're talking about the bear-proof garbage containers have caused natural selection of a higher IQ. This may seem funny, but it's actually derivative of some of their arguments. In some of the journals, you can actually find this sort of level of proposal of things that happen, because they can just throw the word natural selection in there and arm wave, and they'll get a pass on the journals. But that's not how the genes are expressed. This is how they're expressed, by epigenetics. And in this case, you see the DNA molecule, and then there's these little yellow tags on the bottom right, and those are methyl groups which sit on top of the DNA, epi for epigenetics, but they do not change the DNA sequence. There's no change to it. If there's no change to it, there's no mutations. If there's no mutations, there's no natural selection. So epigenetics basically dismisses natural selection on that level. There again is the nucleosomes that you saw in the very first slide. They're like yo-yos winding up the string, and as they tighten, they do away with any random mutations. They reduce them by 90%. So again, the concept of random mutations leading to natural selection does not work as we're discovering now. There's also, on the bottom left, you see these microRNAs. Those are RNAs that are transcribed by the largest part of our DNA called the junk DNA. And we basically ignored this DNA. And we'll discuss it in a few slides in the future. As you can see, the DNA, as you go across the top and to the left, is wrapped up into chromosomes. So it has, epigenetics has everything to do with how the DNA is wrapped up tightly, and how it's wrapped up tightly has everything to do with how it's expressed. This is completely different than the simplified central dogma theory. The central dogma doesn't take into account these methyl groups. It doesn't take into account these nucleosomes. It doesn't take into account the folding of the DNA chromosomes, nor does it take into account microRNA. It in fact ignored microRNA. Once again, this is a GIF showing a summary of how the DNA is wrapped up into nucleosomes and how it tightens up. Now, keep in mind that we have 2 meters of DNA in every cell of our 40 trillion cells. If you unwrapped it all and put it end to end, it would go to the sun and back 70 times. So how does this DNA not break or get destroyed? It's because of the epigenetic molecules that are sitting on top of it and wrapping it to keep it safe. But in its wrapping, it also controls its expression, and which is again outside of mutations and natural selection. So on the right, our approach had been in the past strictly genetics, looking at the DNA sequence and the variations, i.e. mutations, and that was supposedly what caused all the gene regulation. However, on the far left, we noticed that the environment is causing these things through epigenetic modifications. And as you can see, there's orange juice and pills and other things which can change the epigenes without changing the DNA. And it controls gene regulation. And ultimately, that's what causes the phenotype or the changed body. Not so much through the DNA sequence, but it's how the DNA sequence is played. So it's not a matter of gene variation through mutations, rather than gene regulation by epigenetic tags without changing the DNA. That's a huge point. This is kind of a broad explanation of things, but in the middle part, there's these different forms of epigenetic processes. There's the little tags that you've seen in the previous slide. The histones, which are those yo-yo things, are modified. The chromatin remodeling, which is your DNA twisting up into chromosomes. And then there's the non-coding junk RNA, which also regulates things. And on the far left, there's all these different things that can modify it, from environmental stress to genetic variation, nutrition, maternal behavior. These are all things that were not thought to be able to change our DNA as we passed it on to children. These were considered to be actively acquired traits during our lifetimes. And according to neo-Darwinism, only the mutations that occurred in our germline, that would be our egg and our sperm, while they're forming, can cause changes and it's through mutations and natural selection. However, epigenetics goes beyond this. And on your far right, you see the different phenotypes that can come from this, like the quality of milk or eggs or reproduction or disease resistance, wool, and development. This was the Darwin's finches, which is where Darwin came up with his concept of evolution, that is changed through slight change with natural selection. And if you look at a map of the different islands in that area, they're of varying size, and they're all relatively close together. And he saw different types of finches, mainly with mainly changes in the beaks. So he came up with this concept that, well, maybe they just kind of varied for reasons he didn't understand, that we didn't have genetics then. He just thought that living beings could just kind of morph with no real explanation as to what the underlying process is. But once it did morph, if it was more suited to survival of the fittest, it would pass on its genes, which included the morphing genes. So again, it seemed to make sense at the time that he would make this possible thing said, but he would never pass this today. If Charles Darwin were to try to pass his paper off based on this, it would be soundly rejected by everybody. But because we did it before genetics, and this seemed to philosophically agree with people, we've been passing it on and trying to keep it relevant. So evolution says that mutations occur, then natural selection fixes the beneficial ones for different forms. However, a study came up, which is titled Epigenetics and the Evolution of Darwin's Finches by Dr. Skinner. And this is about 6 years ago, which completely turned this up on its head. This was a paper that resonated through the biological community. He basically studied the epigenetic tags, which we had seen in the previous slides, those little yellow methyl groups that sit on top of the DNA. And so he said this study was designed to compare epigenetic changes. There's no changes in the DNA sequence amongst several closely related species of Darwin's finches, a well-known example of adaptive radiation. Now, adaptive radiation is a term for when birds or any living being gets into certain niches, like small little islands, and then they form into other forms that are walled off in a sexual fashion to where they will not reintroduce to the general population. In any event, which is basically how we feel like the evolutionists felt like the majority of evolution occurs. Anyway, he goes on to say epigenetic changes contribute to the molecular basis of the evolution of Darwin's finches. I mean, there it is. And in the paper, they explain that when you look at the changes in the finches, especially in the areas of like bone growth and the beak, that there were more epigenes involved in those genes than there were mutations, which was the Darwin's approach. So the mutations did not line up with the changes in the beaks and the morphology of the finches, as well as epigenetics did. So that was, that's a major thing right there. I mean, that is it, in the sense that for 170 years, we've been following Darwin's theory of natural selection, and it was based on his epiphany in the Galapagos Islands. And yet the finches that he idolized are replaced as one of the icons of evolution, has been disproven, or it's been shown to be evolved not by Darwinian mechanisms, rather epigenetics. So again, here we are with a DNA strand going around the nucleosomes, and you can see that the nucleosomes at the very bottom tighten up with the chromatin compaction. When this occurs, the mutations are not occurring; they're reduced remarkably. So there's no random mutations. There's no natural selection occurring. And also they're not transcribing things. This kind of explains, for instance, if you get the epigenetic tags and they can mutate, the tags that can mutate, not the DNA, but the tags, they can turn off, say, a gene that should have been there to help regulate to avoid getting cancer. And that's what can happen in certain instances, with cigarette smoke and other things may turn off a gene that was needed and subsequently cancer popped out. Or on the other hand, maybe a gene that is associated by being turned on that can cause factors that can cause cancer. So this turning on and turning off is a very sensitively controlled thing. And this is how cancer and diseases are being expressed. So it's not mutations that's causing these things. There are some diseases that are related to mutations, but they're very rare. Less than 1% of the time does a mutation cause a disease. The vast majority, over 99%, of these are caused by the regulation through epigenetics. So again, on the far right, the methyl epi tags tighten and reduce mutations where there's no random mutations or natural selection. Again, natural selection can only occur on mutations. It doesn't occur on epigenetics because there's no mutations or change in the DNA. So these are the different components of epigenetics. We've discussed, the top left, you have the little tags that sit on top of the DNA on what's called the cytosine, the Cs, and they tend to make the DNA tighten up in the balls on the top right, the nucleosomes. If you have a lot of those tags, then that will tighten it up. Now, those balls also have these little tails on it that you see in the top right, and you can put epigenetic tags on those that cause a lot of changes. In fact, I was reading an article that showed a rare form of brain cancer that was very bad for young people. It's all due to one misplaced epigenetic tag on one of those tails. It's very rare, but just that can show you how one misplaced tag can do a great deal of harm. So on the bottom right, we also see that there's the RNA, which also jumps in there to cause modifications of this process. And you can see that also to the bottom left with the non-coding junk RNAs, which are modifying things and telling the end product messenger RNA what to do. Now, if you've had a vaccine, by the way, the vaccine would have been what's in the middle bottom there, what's called the messenger RNA, and it produced proteins from the COVID viral capsid, which then caused your body's immune system to react to it. So that's messenger RNA. Well, let's get back to that subject of junk DNA, because this is really a huge issue. The evolutionary theory predicted that 98% of our DNA was basically junk DNA and was not really worth studying. I mean, that's an incredible statement to think that. And I'm going to show a quick little YouTube to show about what junk DNA is.
In 2001, researchers of the Human Genome Project determined that only 2% of human DNA are genes, the sequences that directly code for the proteins our cells use to carry out their functions. The remaining 98% of the genome was concluded to be a biological wasteland. But it turns out that this supposed junk DNA is crucial for controlling gene activity, often acting as switches to turn genes on and off. These switches determine where and when a gene turns on. When a regulatory molecule binds with its corresponding switch on the DNA, it will cause the switch to flip and the gene to be activated. With the regulatory molecule bound to the switch, the gene will be expressed in greater amounts. When the regulatory molecule detaches from its switch, gene expression will stop. Typically, the gene will be surrounded by many switches that allow the gene to be turned on and off under the control of different regulatory molecules.
Okay, that was rather simplistic, but it did get the point across that due to evolutionary thinking, we thought that this was junk. This is a Science Journal, which is a major one, and it's talking about this project called ENCODE, which came along and studied the junk DNA, and it basically wrote a eulogy for the junk DNA because everyone realized, and this was exactly 10 years ago, that that term was completely wrong. Junk DNA was the greatest argument against creationism, according to Richard Dawkins. And there's a YouTube that shows that. He's saying, if there's this junk DNA in there, that God must have made junk just to confuse Christians. Francis Collins, the head of NIH, who's a Christian, also made this argument, not against Christians, but creationists in general. And as a result, it's rather funny, but sad at the same time that his study called the Human Genome Project was a complete failure because of his commitment to neo-Darwinism. In fact, later, after he failed and the ENCODE project came out to show just how bad he failed, he threw in the towel. He said, in terms of junk DNA, nobody uses that anymore. I think it was pretty much a case of hubris. Isn't that amazing? Hubris, to imagine that we would dispense with any part of the genome. He basically dispensed with 98%. And now he realizes it's hubris. As we now know enough to say as if to know that we could say it wasn't functional, most of the genome that we used to think was there for spacer turns out to be doing stuff. Now, he uses the word spacer. He was calling it refuge. And in his first book, which talked about God and DNA, etc., he was calling it refuge and junk, and used this argument against fellow Christians who believe in creationism apart from evolution. He later went in and ripped that out of his book. So here's a scientist, and when a scientist makes a statement, it's supposed to stay. You let it sit there in the literature, and somebody else should disprove it. He instead snuck back there and pulled it out of his book because he was ashamed of the idea that he came up with and that was wrong. He's still an evolutionist in that sense, but he's not going to admit that it really caused us a lot of problems. So on the far left, we have the central dogma, which goes DNA to RNA to protein, which is a very simplistic way of looking at things. And that was neo-Darwinism. But on the right, we have these non-coding regions that go between in the DNA that pepper it, like you saw in the film. And after it's spliced out, it does all kinds of changes in the folding and translation and forming new genes. These are some of the junk DNAs that form into non-coding RNAs, and there's a whole variety of them. And the combinations of the things they can do give, by one researcher's estimate, an almost infinite number of possibilities. But how did a new gene come into existence? I mean, how do we actually form a new small gene? And on the far left, you can see John Lennon. I use that image because he says, imagine. We've all heard that song. Well, in the article on the right, it's called Darwinian Alchemy. Darwinian Alchemy. They don't sit there and say, we're going to show you how a new gene forms. They evoke alchemy, kind of your imagination of human genes from non-coding DNA. That's the junk DNA. And they say one can imagine the final phrase of this article. they conclude, how could all these pieces of junk DNA fall into place through the random processes of mutation? There's their mutation, they're still stuck on it, right? Now, one thing to point out, when a mutation occurs in the junk DNA, by definition, junk DNA does not directly code for protein. And according to the modern, to the central dogma, you have to have DNA to RNA to protein to have a better being, therefore fixing the mutation. Junk DNA doesn't code for protein, therefore it's neutral. It's considered completely neutral. So there is no mutation in natural selection. So they say again, random processes of mutation, recombination, because this junk DNA is shuffling all around, and neutral drift. This is where chunks of junk DNA will move from one individual to another or population, or at least enough of these pieces to produce a protogene. This is a protogene, is the very first beginning of a gene that was sufficiently useful for natural selection, and that's Darwin's natural selection, to take hold. Now, that is an incredible, significant statement. You have to have a gene for natural selection to take hold. Well, how do you derive that gene? If natural selection is not causing it and mutations are not causing it, how do we get that? They don't. Darwin discusses the arrival of the fittest, the survival of the fittest, not the arrival of the fittest. And that was a statement made 100 years ago. So genes, they just discover that genes show up, and they don't know why they showed up. But look at the very bottom. One can imagine, he said, one can imagine a small gene arising and it getting larger. That was his conclusion of this journal article. This is what is passed off as journal acumen, to just imagine the thing occurring. They admit that you can't explain how you can shuffle junk DNA to form one, because it's not under selection, or if it did come along, how could it get to the point where natural selection could take a hold of it? So it's a rather remarkable. Now, this article I found out of using Google Scholar. I did a search on natural selection. There was 3.5 million journal articles that have those words in it. And then I did a search on de novo genes, which is what this is about. 90 papers popped out of 3.5 million descriptions of Darwin's natural selection. Only 90 papers popped out, and this was the only one that came even close to explaining how a new gene came into existence. So out of 3.5 million journal articles, one article about how a gene comes out, and it basically begs the question, how could this have happened? Because natural selection couldn't have got a hold of it unless you already had the gene. And then the final conclusion, we can imagine one coming out and getting more complex. Isn't that amazing? At the end of the day, decades of research, millions of journal articles, and this is what we have. So this is, of course, Stop the Press. We're talking about random mutations, etc. And this was a journal article that came out just within the last few weeks. In the first of the article, it said random mutations are an important part of the theory of evolution by natural selection, in which mutations give rise to adaptations that are passed on to offspring and alter their chances of survival. You remember the penguins jumping up on the ice. They had a mutation, they got a bit stronger, and the stronger penguins are passing their genes on. This article goes on to say, since the first half of the 20th century, evolutionary theory has been dominated by the idea that mutations occur randomly with respect to their consequences. So 100 years. They concluded in their paper that epigenome, the epigenetics associated mutations, reduce the occurrence of deleterious mutations in the Rabidosis. It's a weed, basically. The Rabidosis thalia is the most studied, basically, weed. I think it's from Australia. It's like Darwin's finches. It's so well studied as a model for epigenetics that, countless articles are coming out using it. But they basically are challenging the prevailing paradigm that mutation is a directionless force in evolution. That is huge. Paradigm, it's a paradigm for 100 years, or really since Darwin, that mutations is a directionless force in evolution. Darwin, the reason people love Darwin is it proposes directionless forces change things. And with directionless forces, you don't need God. And so random mutations is supposedly you don't need God because random mutations made us what we are. It's the goo to you by way of the zoo, basically. They all just mutate along and directionlessly formed with what we see. This statement right here means that this 100-year doctrine of random mutations, which stood in the door and pushed Christianity back and said that we're just a directionless force in evolution, has been disproven. That is an incredible statement to say that this has been disproven. Our discovery yields a new account of the forces driving the patterns of natural variation, challenging a long-standing paradigm. There it is, a 100-year paradigm regarding the randomness of mutation, and inspiring future directions for theoretical and practical research on the mutations. Basically, they're saying, look, guys, we better look into this because this is what's happening. If you can find one example, and this is not really the first one, there's been a lot of these papers coming out over the last 20 years showing that mutations is not random at all. So basically, here we are on the top left looking for those random mutations. And I don't know if probably none of you were alive when this commercial was out, but it was hilarious and very funny where these three old ladies would drive up to the drive-through for Wendy's hamburgers or any hamburger place, and they said, "Where's the beef?" Wendy's had the beef, but the other ones didn't. So they're basically saying to the evolutionists who believe in random mutations, "Where's the mutations?" Backing up to that slide at the very first, which basically is a summary of neo-Darwinism, says you have DNA goes to RNA, goes to protein, which is the central dogma, but the mutations happen up in the DNA, and that's passed on. And it makes different protein, and they have a bigger muscle, or they're the penguin that can jump higher, or they're the bear that can now suddenly figure out how to get past the garbage can and evolve. But that's not the way it works. This is the way it works. There are these yellow tags called methylated DNA, which do not change the DNA sequence. There's no mutations. They sit on top of the DNA. They make it fold around the yo-yo shaped nucleosomes. They modify the histones tags, which can cause tremendous change. They control the way the DNA is highly compact, and they work with the junk DNA, which forms micro DNA on the bottom left. This is just a simple illustration or GIF of how things work. One piece of DNA with different tags, epigenetic tags, can make different types of cells forming on the bottom. As you see it, the same DNA, it's making these different forms. Again, different forms from the same piece of DNA, all by the epigenetic tags, not by mutations, not by natural selection. This is a diagram made by Conrad Waddington, who was the gentleman that coined the term epigenetics back in the 40s of last century. I mean, a lot of people have been telling the general evolutionists that something is not working, and they would not listen. They would just say it's one-offs or something. So it's really sad that their commitment to that theory really set us back. I mean, at least 50 years in medicine, we've been set back because of this. Okay, this is the article called Epigenetics and Making of the Ant Casts. Okay, we saw on the previous slide that you can get all kinds of different forms with one piece of DNA. Okay, well, the same thing applies in the social insects. From the same DNA, you can have entirely different phenotypes. That's bodies. You can have soldiers, common laborers, and queens. And they've discovered that this is all due to methylization, i.e. epigenetics. This was a complete paradox for Darwin as well. and Dawkins. There was a very strong debate going on with Richard Dawkins and E.O. Wilson of Harvard, who recently died, and Wilson was believed in Lamarckian epigenetics. Dawkins didn't, and they were almost in fistfights over this. But he said, how can you explain these different forms that you see in the bottom left from the same DNA unless there's some form of changing epigenetic tags on the top? And you can see the ball rolling down on the top left, rolling down the different valleys, and that's the epigenetic paths that it takes. So the ball can go in many different paths and form many different forms. Now, this situation is so bad, basically, that as far as epigenetics and what it's causing, that this article says the field of epigenetics is causing a major disruption in the field of evolution. That's a very important statement. Creationists are not causing any problems for evolutionists. They just dismiss us outright. Nor is intelligent design. Stephen Myers has done a great job, and I would say Darwin's Doubt book probably did, caused a lot of stuff, but everyone just kind of ignored it and accepted the fact that we can't explain the Cambrian. So, and he defended it very well, but it didn't change anybody. Epigenetics is changing everybody. There's at least 100,000 articles written on this in the last 10 years. And it's changing everything in a major way. So a new recent news article reports that current estimates are approximately 1/3 of professional academic biologists, and I would say that's more than that now, who do not believe in intelligent design, so that they're not creationists per se, find Darwin's theories inadequate to describe all the complexities of biology. So this is, epigenetics is the way to go, not the older Darwinian arguments. This is a book I've been reading called Lamarck's Revenge by a paleontologist, and he's basically blowing up all the older theories because he realized the Cambrian could not be explained by Darwin. And he said today a revival of Lamarckism, which is another word for epigenetics. It was Lamarck was around 50 years before Darwin, and he proposed that the environment causes change. Basically, people ignored him for 200 years practically. But it's causing many biologists to admit that a major revolution in evolutionary theory might be at hand. He goes on, but on occasion, even seemingly excuse me. Even seemingly irrefutable scientific principles. This is talking about neo-Darwinism. Paradigms do falter because of revolutionary new discoveries that cannot be explained by the old theories. That is an incredible paragraph. He's basically saying that neo-Darwinism is incomplete. It doesn't explain things. And we have this new revolutionary discovery that does explain things. So, and there we were believing that this was an irrefutable scientific principle. That's what, when I was in school, that's what they were teaching, that this was irrefutable. There's no way to get around it. And they did not understand epigenetics. There had been hints of it in the literature, but they basically ignored it. So anyway, that's a quick summary of epigenetics. And I have another YouTube, which can follow on this. If you go to YouTube and look up my name in epigenetics, I did a 4-hour lecture, which goes into a lot more detail. So till next time, we'll see you then.